thank you for the organization of this satellite event here to Ibro. Yeah, it's always it's difficult to organize an event, to put together specialists from abroad to share with us and to enrich the environment of the Brazilian neuroscience. And would expect that we should always make the news in terms of these kind of events that are expensive and effortful, to have more people to put our students. So as a Brazilian neuroscientist, I see that people from Ibro that are not here lost very good time. So just going to, I'll try to not to be too long. My history comes from synaptic plasticity, where it raised in me the interest in studying the communication of hippocampus outputs by the time where most of the studies were focused on interaction to hippocampus communication. So the other output that at some point I came out to see was that this description, this anatomy showing that the hippocampus projects to the prefrontal cortex. So very few studies had started at that time, and it's great to see that many recent studies have been focused on coupling and interactions, so we could understand better how behavior is being controlled by these circuits. Today I'm going to present here quickly a new line of interest that I started when I moved to Natal, was to try to apply the strategies of multi-unit recordings and LFP analysis and also plasticity to study a circuit that is involved in comorbidity that happens with epilepsy. That comes to be autism. It's people with autism has frequently have epileptic seizures, 30% of them, and around 80 to 90% of the children that has autism has changes and aberrations in the EEG. So that was actually the link that made me to go from studying epilepsy to why autism. I'm not going to talk about autism by itself. I'm going to talk about a little bit of the behavioral and how can we study some of the endophenotype associated with autism. So social behavior you can describe as species-specific adaptive behavior. So it's from insects to mammals. You have these behaviors. It increases the survival reproduction probability, improves safety perception and affection, and the welfare state of the animals, and modulates behavior strategies. So every animal has to design and to come with behavior strategies to execute their everyday life. So social behavior is a complex behavior that involves sensory inputs and motor coordination, sensory motor coordination, and other aspects. And impairments of social behavior is associated with several disorders, and not only with disorders, but moments in an animal's life or a person's life. So here you can see that these social behaviors are in insects, but in the rodents that we most frequently use to do neuroscience, it's very common to have when they're young, when they're teenagers, let's say, they have this behavior of play and tumble, and when they have other behaviors that make them to socialize when they are in the wild, they also reproduce these kind of behaviors. So what are the neurosystems involved in social behavior? So you're going to a very complex, and that's not the way usually you learn how to do science. You learn how to do science reducing to simple questions and ask each one at a time, right? So, but at the same time, you want to understand things that are closer to you. So there are different ways of doing that. So you can split, modalities of what you call social behavior. And you can pinpoint and choose one of these modalities and select one to study. So this is one way of doing that. So social behavior involves sensory perception. So you have odor. In rodents, it's very used, but the auditory also plays a role, as rodents emit in ultrasound, also emit in the audible frequency. But motivation state is important to social behavior, as well as sensory motor coordination. So animals have to modulate their behavior according to the partner's behavior. And cognitive processing too. You have time processing for coordinate behavior, as well as the affective state and the emotional state of the animal. So this is a multi-level process. When you test animals and in social behavior tests, all these modalities are actually combined and being used at the same time. So just to give a quick look in terms of what is the relation of the model that I'm going to talk about with what you see in autism. Autism is a developmental disorder, so something is happening during development. But it's very heterogeneous. It's very hard to go into the literature and to make sense of different studies because they have a number of subjects very small, and many times it's very heterogeneous. So it's hard. You have to be careful. And there are aspects that are genetic and other aspects that are environmental. Here today we're going to talk about environmental that is basically responsible for most cases associated with autism. And several descriptions, several classes of what is combined that has these different deficits associated with social skills, restricted interests, and repetitive behaviors. But if you read very carefully, there are a lot of sensory integration abnormalities in these people, in these children. So, more recently, several studies have pointed that the prefrontal cortex and other areas are altered in children with epilepsy, with autism, and in this case here in this study, they collect several specimens of prefrontal cortex, and they evaluated that there was an increase in the number of cells in the prefrontal cortex of children with autism. Other studies have been shown with event-related potentials that this is also the case. And auditory-visual integration in response to pulses and visual stimuli are also affected in autism. So, what brings me to this field? Well, I wanted to try to understand how changes in temporal cortical circuits could affect a behavior that involves the interaction with more than one animal. So I came up to this model that is called the valproate model of autism. And it comes from the literature where women that are taking antiepileptic drug, that is valproic acid, they have an increase in 10 times the chance of having children with autism. Differently from what is seen for general autism, where the sex ratio, male to female, is 4:1, in the valproate syndrome, the male to female ratio, it's 1.2:1. Very different from what you see, probably because of the heterogeneity. And so the description of the effect of valproate is being long in the literature, but more recently this study collected 600,000 cases of autism in Denmark. And so they actually showed that there is a increased risk. In 1996, Rodrigo in France developed this model where she injected valproate in rodents. And these rodents had a decreased number in cells in the brainstem and had some deficits in cells that were lost in the cerebellum. there were, at the time, was a landmark for autism in terms of neuropathology. So what is valproate doing? Valproate is inhibitor of the histone deacetylase. That is, it's an enzyme that takes out acetyl groups from the DNA, from the histones that pack the DNA. So making the chromatin to stay in this form rather than going to this form that is where gene expression is repressed. So valproate tends to keep the chromatin in a de-repressed state where gene expression can be released in the heterochromatin. But VPA also has the effect of increasing the GABAergic transmission, and also is, increasing the release of GABA. So this was the original paper showing that some abnormalities in the animals injected with valproate, and recently it was characterized that these animals have some behavior abnormalities associated with lack of nociception, reduced nociception. They have lack of social interaction. So this is basically what I've said. Valproic acid is produced from this plant, and there are some analogs of valproic acid that keeps only the acetylase action, but has no GABAergic effect. So in that case, those drugs don't produce the same effect in the offspring. So it's, even though other antiepileptic drugs can induce states of malformations if given in a very, very high dose during pregnancy, valproic acid seems to be working through its action in gene expression. So to generate this model, we used to alter gene expression through valproate. We suppose we're acting through gene expression when the rats are pregnant. So we inject at the time when the rats are pregnant, and you affect some of the processes that are going on during development, and you can see the changes and the activity when the animals are born and later on. If you basically see the animals, they are not very different except for a phenotype trace that they have in the tail. I'm going to show you. I'll just jump it quickly. Recently, some experiments have shown that the GABAergic system in VPA animals could be affected. And I just brought this figure here to show the diversity of some of the GABAergic subtypes of cells that are seen in the cortex. This is not all of them, but these are the majority of them: parvalbumin, somatostatin, and carotenoid. And these cells are distributed in different layers, and the pattern of expression of parvalbumin, for example, is time-dependent, so it's developmentally controlled after birth. These cells, they have birth dates that are different during development. And by the time of the injection, you probably should be affecting the pool of these cells. It's one possibility. So we decided to see if we could reproduce this model and try to study these animals in the lab, trying to focus in the hippocampus prefrontal cortex circuit to see if inhibiting that communication could somehow affect their behavior. So basically, you treat the animals, the pregnant rats, at E12 with valproic acid. The animals are born, and you test them in behavior tests, and then you can implant the animals and check other histological patterns. But in our way of doing this, we generated these animals, we test. I'm going to show you the results. We had a box, a cage, where the animals mated among themselves, VPA with VPA animals. And we had this offspring of the F1 animals. So we decided to go and to compare these two groups and in terms of behavior. That was actually an accident. We didn't expect, we didn't go that direction. But in the end, it took us to have some interesting findings that I'm going to show you. So here you have an animal that was prenatally treated with VPA. You can see, or not well see here, you have a kink in the tail. The F1 doesn't have a kink in the tail because F1 wasn't exposed to VPA. But what you see is that the VPA-treated animals they don't gain weight as the regular animal, but the VPA, the F1 has a weight gain normal. Doesn't seem to have any difference. However, the eye opening is one of the aspects that you can look for developmental postnatal development, and you can see that the VPA and the VPA, they don't, the fraction of the animals that have the eyes opened change along the days. So most of the control animals have their eyes opened by P13. So compared to VPA, and VPA animals, they take longer to open their eyes. That had already been described for the VPA animals, but not for the VPA. Here, we show that, for example, the animals have hyperlocomotion, the VPAs. However, and the VPAs has a trend to have hyperlocomotion. The VPA animals are more anxious. They spend more time in the periphery, close to the walls, compared to the center, while the VPA animals have a tendency to go in the same direction. There are some differences also in exploration aspects of the cage and grooming, where the VPA animals are more prone to have repetitive behaviors compared to the VPAs. Here, we did a regular test that is to expose the animal to a 3-chamber classic test of social interaction, where you keep a strange animal here, your experimental animal is put in the center, you allow the animal to explore all 3 compartments. So you measure basically the time spent in each compartment, and you have an idea of how much this animal has the tendency to stay closer and explore a new animal as compared to just a regular box, a regular cage as an object. So if you see these are the absolute numbers, you see that the control animals, they tend to spend more time in the social chamber where another animal is, while the VPA spends similar, equal times in both chambers. And the VPA animals, the F1, the F2, sorry, they have a phenotype that is actually in the middle. So this we did in we did in 5 to 7 liters of each, just to show that that was an effect that could be reproduced independently of the litter. So this is something that came up to us, and we said, okay, so that should have something interesting in terms of what's going on with these animals. So these animals are in the midpoint in terms of that behavior. Let's see. So if you look at the cortex of these animals, you don't see, you're not going to see much of a difference. Here is just to show the areas that we're focusing in terms of the recordings and also the histology. So you see that there is a cingulate area here. This is the motor cortex over there. This is the olfactory bulb. Here is the prelimbic and the infralimbic, where the hippocampus projects strongly to this area here, especially for the deep layers. Can we... The lights can go down more? I don't think so, right? Okay. So, so these, we just, as we wanted to do the recordings in these animals, we wanted to, just before going for the recordings, we wanted to see the state of a specific inhibitory circuit there in the prefrontal cortex. So we just labeled for a subgroup of cells that comprise 40% of the inhibitory cells in the cortex. That these cells are labeled with parvalbumin. Parvalbumin is a calcium-binding protein, and these cells are basket cells that innervate the cell body and proximal dendrites. And there are also cells here that label for parvalbumin that inhibits the initial segment of the axon and has a very strong inhibitory power. And so these are the cells. They have parvalbumin very strongly in the cell body, but they form a plexus. And this plexus are where the axons of these cells are actually inhibiting their targets. So we just checked if parvalbumin that we measured was co-labeling GAD65, that is a marker of GABAergic cells, just to be sure that our cells were really GABAergic and not unspecific parvalbumin cells. And what we showed was that if we just grouped the data, we saw that the number of parvalbumin cells here in controls and in the VPA animals. So we had a reduced number of parvalbumin cells in the prefrontal cortex of these animals. Comparing to the VPA animals, we saw actually an increase in the number of cells in the prefrontal cortex. And so if you divided, when we split the data into the subareas of the prefrontal cortex, we saw that that was concentrated in the cingulate cortex and also in the prelimbic specifically, especially for the difference in the VPA. So if we look back and look at the behavioral results, that we didn't see actually a phenocopy of what happened with the VPA in the VPA. Here we see the same. There is not a direct reproduction of what happened with the animals that are directly exposed to the VPA when you analyze the animals that are their offspring. So we are at the point that from these results, with these results, we are thinking that, okay, now I have this decreased number of parvalbumin cells in the prefrontal cortex. Sudhoff group had recently shown that in the somatosensory cortex of animals treated with VPA, there was also a reduction in the somatosensory cortex that could somehow point to a behavior that could be associated with the lack of sensory processing or a difficulty due to hyperexcitability, local excitability in the somatosensory cortex. So here is just the gross figure of picture of the prefrontal cortex in a control animal. In a VPA, you don't see major difference in the prefrontal cortex. So this difference seemed to be more subtler than we would expect to see from an animal that was prenatally exposed to that drug. Markram's group they published a paper a couple of years ago where they pair-recorded pyramidal cells in the prefrontal cortex of VPA-treated animals. Their thinking was that, based on some previous hypotheses on autism, that the hyperconnectivity was associated in autistic kids with local hyperconnectivity was associated with the weaker long-range connectivity. So this hypothesis has kind of leading, many researchers to design experiments trying to test it. And at the time, what they showed was that pair-recording pyramidal cells in the prefrontal cortex, the frequency of pair-recorded cells in animals that were treated was higher as compared to controls. They did this separating actually what they considered short-range connections to long-range connections. They considered 100 and 200 micrometers to be short-term and long-term connections. I think that further studies should address this question using longer distance and checking better, this connectivity in other ranges. But anyway, I think this data pointed to a place where you could go with other questions. So what we've been doing in Natal is to set up a set of experiments where the question is very simple at the beginning, is to see how is the state of the hippocampal to prefrontal coupling in these animals. First, at just a regular behaving task where the animal just stays exploring, where I can measure theta, where I can measure Leah, where the animal is just quiet, drowsy, or in a state where I can see oscillatory events that are already traces or marks of hippocampus to prefrontal coupling. So, but from our recent results, our recent data, I can see that these animals have some oscillatory patterns that sometimes seems strange. When they have this kind of spike discharge that are around 7 hertz, that are not actually theta rhythms, are not associated with walking, are not associated with REM sleep. They just show up. This has been shown in some papers recently that can be associated with the age of the animal. And some animals can show more of these or with the strain of the animal. So, so our design is we'll record it at some point in an arena, just in a round arena with a Neuralink system where we record it from the hippocampus and prefrontal cortex. Now we're moving to trying to set up the recordings using the INTAM system and trying to make it in a way that we can record during behavior interaction, this animal with other animals, actually with one other animal during a social interaction task. Here, I just show you a print screen of some of the events that sometimes happens when these animals are much more prone to, much more sensitive actually to be induced epileptic seizures. The same dose that you use to induce seizures in a regular Wistar animal, you can use 1/3 of that dose, and you induce the same kind of seizures and have the animals to present these sporadic events that you see here spontaneously during the lights off period that you see these events. And if you just zoom in these events, these are regular events. They're usually high amplitude events, and they have a signature if you're recording different areas of both hippocampi. And we are actually going after this and try to see if it's just a regular thing that it's with an epileptic animal or it has some different aspect in these animals. So we've designed this kind of apparatus where we are actually doing our experiments, where we have this T kind of box where you have an animal here, a strange animal, our experimental animal. You have here an object, and you have zones defined in this arena. You have an ultrasound microphone here, an ultrasound microphone here. These microphones are connected and synchronized through a National Instruments board, and we are recording from this animal and trying to get a closer look of what's going on in this animal during the social interaction or just a regular behavior here in this arena. So this is a regular trace of the ultrasound emitted by a control animal, and this is an ultrasound emission. This here goes from 20 kilohertz to 90 kilohertz. They emit different, usually different bands, different kinds of actually ultrasound emissions, usually 20 kilohertz and 50 to 70 kilohertz. So this is one group of experiments that I have some students still collecting some data. I don't have more data of the electrophysiology I would like to show here, but this experiment that I'm going to show you was trying to understand if these animals were actually, how was the sensory perception in these animals. So we had a collaboration with a researcher from McGill University, Dr. Etienne Viesa Zidane, and one of our students went there to record from the auditory cortex, A1, primary auditory cortex, to check if these animals had some changes associated or not with the deficit in the parvalbumin cells. So for these experiments, our question was simple. Were the neurons from the primary auditory cortex responding differently to sound beeps, regular electrophysiological recordings from A1 as compared to the controls? And if so, would that be related to the number of the organization of inhibitory circuitry inside the auditory cortex by measuring the number and the distribution of parvalbumin neurons in the auditory cortex? So, so here is where the recordings were done in the primary auditory cortex in rats. The auditory cortex is divided in 4 areas. Actually, the primary auditory cortex and the secondary supplementary auditory cortex. 2 of them have a tonotopic map, and this tonotopic map is organized tail to tail. So these are kind of mirror of each other, these maps. But there are clear borders to define each of these maps, especially based on anatomical response, the latency of the responses. So these recordings were done using a 64-channel array centered into 8x8 matrix that actually covered more than the primary auditory cortex. So the array passed the border of the auditory cortex. So we could define where neurons finished or didn't respond. So the sound was presented. So this is just an example of a response. Neurons that responded to a sound beep. We range from 1 kilohertz to 60 kilohertz, pure tones with steps of 0.5 kilohertz from 3 to 70 dB. Sorry, from 0 to 70 dB. We did these number of animals, and the animals were at adolescent age. So what we saw here was that this is a A1 map for a control animal. Usually, it's been repeatedly shown in the literature that the tonotopic maps in rats, it's described from a rostral to caudal, a representation of the high-pitched sounds to low-pitched sounds. So the higher frequencies in the ultrasound range are represented more rostrally, and the lower frequencies are represented more caudally. So here you see these maps, and where the axes appear is where the cells don't respond, the cortex don't respond anymore. And some of the spots, sometimes you may have loss or non-responding areas too. So here you see, if you normalize by the tonotopic axis, anterior-posterior axis, you see that the highest frequencies are more organized in the rostral and the lower frequencies in the caudal part. As compared to the controls, you see that in the VPA animals, you see a kind of a mix. There are some sites that respond to lower frequencies more rostrally, and sites that respond to higher frequencies more caudally, as if the map was kind of spread all over the axis. This is just an example of one animal. I'm going to show you in the next slide an analysis of the group. The other way you can analyze is to check the tune curves of these animals. The tune curves of the control animals are usually, if you are in A1, are usually sharp, where you have a very specific sound frequency, the specific sound frequency, where the cells respond to the lowest sound intensity. By the time you increase intensity, this range of response, they increase, and you can also quantify how wide is this V shape. The wider is this V shape, it means less specific is that neuron or that site. So usually in the controls in A1, you have most of the sites are V-shaped, where cells are tuned to one frequency. However, in VPA animals, you have sites that are V-shaped, but you also have more sites than usual than the V-shaped sites as a flat sites, where quickly they get in a specific, just by increasing the intensity. So here, if we quantified it, we show that this is sound, the sound frequencies. Actually, this is hertz, this is not dB. And the percentage of sites that respond to at these frequencies. So in VPA animals, you have more sites responsive. Sorry, here is yeah. This is 50 dBs, that's right. So here it shows actually that the animals have more sites that are only responsive at higher intensities of the sound at their specific frequency. So this here, the specific frequency was determined for that site and used at that site at different intensities. So these animals are kind of, from this graph, we interpret that these animals are kind of deaf, or a bit deaf. They need higher intensities to respond. Then there are more sites that are only responsive at these intensities. On the other hand, if you compare, for example, the number of V-shaped sites, well-tuned sites, to multi-peaked or flat sites, you have a decreased number of V-shaped sites and increased number of flat sites in VPA animals, showing that these animals have worse tuned A1, as if the primary auditory cord was less tuned to their specific frequencies. This is another way to show that the V-shaped sites are wider. You measure this, the frequency range 10 decibels above the minimum intensity of response. And here you see percentage of high threshold sites. So this is higher. However, the latencies of response are lower. So these animals have an impairment in auditory processing, and we saw those differences in the parvalbumin number of cells in the prefrontal cortex. Are these cells also altered in the primary? We saw that in the prefrontal cortex. Are these cells also somehow decrease in number in the auditory cortex? So we measured it, we didn't see anything, and there was kind of a surprise, but we didn't see any difference in that specific number of cells. Of course, that there are other possibilities, as number of synapses and other internal connectivities or inputs or thalamic inputs into the primary auditory cortex. So I'd like to to thank, my students that did part of this job. And as I mentioned, the way we're going now is to do the behavior recordings in those animals and trying to figure out how the responses are altered when the animals actually get closer to another animal, and if that is related to the ultrasound emissions. Thank you.